Hello, everyone. Thanks for joining us virtually for Michigan Mushrooms with Legacy Polycultures. And following along as all of our programming has changed from live and in-person to online, we hope that you'll enjoy this presentation. And let's welcome Andrew. Thank you for being here with us today.
Thank you. Thank you, Ms. Sarah. This is Andrew Mateskon. I owner and operator of Legacy Polycultures. And I'm joining you from the Nelson Township Library, the beautiful Bill and Virginia Vanderkellen study room.
Wonderful. I appreciate you helping us get this information out to the public. It was a very popular program, and we had a lot of people asking, can we still hear what you have to say? So I'm going to turn this over to you and let you share your screen.
Sure. Thanks. Yeah, I'll dive right into the presentation then. Just a moment while I share my screen. Okay. So today we're going to be talking about mushrooms of Michigan. These are some popular mushrooms and some mushrooms that I've collected this year on your screen. Yeah, let's get into it. So I am not a trained mycologist. I'm an amateur, but I love mushrooms, and that's why I'm presenting on it and like to hunt them, forage them. Professionally, I'm a estate planning and agricultural law attorney, but mushrooms are a hobby of mine. I like to photograph, go on forays, walk really, really slow through the woods. Finding mushrooms takes a long time when you're walking, and I like to learn more about them and share that. So that's why I'm here. Before we get started, I want to just read this slide off. I know I'm not supposed to do that as a presenter, but I just want you to know that I'm not an expert. I don't consider myself an expert. I'm an amateur, and I love mushrooms, but that's where I'm at. This presentation is for educational purposes only. I don't give advice on edibility, medicinal use, or ID, because I'm not present to identify whatever you have in your hand or whatever you find in the field. I think mushrooms are interesting, even if you're not in it, just to eat them and potentially poison yourself, or potentially have a really delicious experience. It's all at your own risk. And then remaining and watching is consent and acknowledgement of this. I also want to say some of these photographs are not mine. They're under fair use, and I attribute the photographer wherever I use them. Okay, so when we talk about mushrooms, a lot of people have their favorite common names of mushrooms, and you'll notice in this presentation I'll use some common names, and then I'll use some Latin binomials. Latin binomials are like the genus and then the species, and you combine them into capitalized G for the genus and lowercase s for the species, or in the example of this mushroom, which is Helvela crispa, capital H, lowercase c. And this is a really fun mushroom to find. I always find it right at the edge of a trail. So it grows in gravel, basically. Mushrooms can grow pretty much anywhere. These like gravel right next to trails. I find them growing together usually, not from the same, spot, but multiple, in a line, sort of how this is. I believe these are edible. They're a fun mushroom to find. It has a common name of saddle mushroom, sometimes called elfin saddle mushroom. Common names often correlate with Latin binomials, but they often don't. So there's Helvela crispa, this mushroom, which is the saddle mushroom, but there's also Serioporus squamosus, Jaramich infula. By the way, if you're using Latin binomials, don't be afraid to butcher the pronunciation because Latin is a dead language. You can pronounce it any way you want. One thing that when you're talking about mushrooms, because the Latin binomials correlate to one species, but the common names can invoke multiple species. Common names are inexact, so you might get a bad ID if someone is giving you common names. So yeah, here's the second one, Serioporus squamosus. This is also called dryad saddle. And then Gyromitra infula, also called elfin saddle. So let's do a little quiz. Normally when I'm presenting, it's in front of people. So this time I can't get audience participation, unfortunately, but let's try to name this. A lot of people call this beefsteak. This is a mushroom that you find in the spring when the morels are up. And as opposed to morels, they call this beefsteak. Sometimes they call it a false morel, but there are multiple false morels. Sometimes, or actually always, this is Jaramich Esculenta. This is a potentially toxic mushroom, but can be made edible with two boilings of water. This one is also called beefsteak. So you can see, let me go back. Oops. Sorry about that. Tried to go back. But you can see that this beefsteak is totally different from this beefsteak. This one is called beefsteak because when you cut it, it bleeds a little bit red sometimes, and it has like a marbled look, like a well marbled steak. It's got white veins and red veins running through it when you cut through it. But the Latin binomial is Fistulina hepatica. So let's keep playing. This is a false morel, Verpaconica. Another false morel, Gyromitra infula. And another false morel, Verpabohemica. So these are three totally different mushrooms. They look totally different, except maybe the two Verpas look similar. But, they all go by false morel as the common name. So I'm just trying to impress on you that common names are inexact. However, common names do have some helpful explanatory power. So they're all false morel. That's telling us that they look like morels, and they do. They look close to a morel mushroom. They're also up and fruiting around the same time that morels are up and fruiting in the spring. So it's a way to differentiate from what people are usually looking for from something that they don't want to look for. I believe all three of these species are edible, even though they are false morels. So it doesn't tell you much about edibility, but just, they're related. And so common names can be useful for that. So, in conclusion on how to talk about mushrooms, Latin binomials can be are exact. Common names are inexact, but can help you understand some general characteristics of the mushroom. Oh, it looks like a morel. It looks like a thing that a woodland creature would sit on, so it's a saddle. So when you're first starting to talk about mushrooms, try to use both. Try to move from common names into the exactness of the Latin binomials. And then I wanted to talk a bit about the deeper history of mushrooms, which is basically a story about life on Earth. Scientists classify life on Earth into domains, then kingdom, phylum, class, order, family, genus, and finally species. When we're talking about mushrooms, we are just basically talking about genus and species. But I wanted to go over some of the interesting things about the kingdom fungi and some of the phylum or phyla as well. Here is a graph showing supposed to be kingdom fungi. You have monokaryotic fungi. There are just a few here, and then Basidiomycota and Ascomycota. And interestingly, right here are the yeasts, and yeasts are in the same phylum as Morels, which are over here, which is something that I did not know. So when we're talking about kingdom fungi, the biggest thing that separates them from other cells or other creatures is that there's chitin in the cell walls. Chitin is a really hard substance. It provides the mushroom with structure, and that's one of the reasons that you have to boil mushrooms to remove certain toxins or to extract medicines from medicinal mushrooms, is that you have to provide enough heat for the chitin to break down. And then there are single-celled fungi, which I talked about a little bit. Yeast is either single-celled or multi-celled, but it's often single-celled. And then you have the multi-celled, and the mushrooms are grouped into ascomycetes and basidiomycetes. So the basidiomycetes make spores using this structure, and then the ascomycetes, it's called a basidia. The ascomycetes make spores using this structure, and it's all, microscopically. So in the ascomycetes, you have the cup fungi, morels, and jellies, but also yeast. And then in the basidiomycetes, you have bracket fungi, typical meadow mushrooms, and boletes, which are pored mushrooms with pores under them. So about 2.4 billion years ago, there was fungi in water, basically, and they weren't terrestrial yet. And aquatic fungi are still with us. Less than 1% of species have been described by most estimates. Some of them are mushroom-producing basidiomycetes. And some of them are, if you looked at our previous slide, some of them are have an ascus or ascomycetes. This is what an ascus would look like for an aquatic fungi. And then around 500 million years ago to 1/3 of a billion years ago, there were, in the fossil record, prototaxites. So these are basically huge mushrooms, as you can see, up to 10 feet tall. They were taller than any of the plants that were around this time. This is before trees, when plants were waist high, maybe, mostly ferns. And this was the spore producing part of the mushroom. They were breaking down vegetable matter, creating soil, breaking down rocks, which we'll get into in a minute. And so I just thought it was interesting that they used to be enormous. I want to talk about the life cycle a little bit as well. Mushroom is the fruiting part. It's like an apple, is the way I like to think of it. The apple has seeds in it. The mushroom has spores. The spores drop from the mushroom, find a place to grow, and then they shoot out this little microscopic filament called hyphae. And then the hyphae join together with other hyphae around it and grow into mycelium. And then the mycelium will, gather food and then produce mushrooms. And we'll get into sclerotia in a minute. But here are some spores. On the left is a spore print. This is where you just take the mushroom that you're looking at, put it on a piece of paper or a piece of foil or on glass, and it drops spores. And it's actually one of the ways to identify mushrooms is to take a spore print, and you can tell by the color, some general idea of what mushroom you're dealing with. If you take a spore print and put it on a microscope, you can get it. You can see what the spores look like. This is under, I think, 100 magnification. And then this is actually a section of gill, I believe. I wanted to zoom in on this a little bit. This is the section of the gills, and you can see the spores being produced and some of them dropping off of that. And then you can look at the shape of the spores if you have a microscope to further identify mushrooms. They come in all different shapes. You're on the level of around 6 to 8 micrometers for these spores, different sizes for all of these, and different shapes and characteristics and colors. I wanted to show you what hyphae looks like under the microscope. You can see it's starting to branch out a little bit. And when these all join together, it'll be more mycelium than hyphae. And then a quick video of hyphal growth and branching. I thought it was interesting, so I wanted to share it. Oh, it's going to repeat. And you can see resources and things inside the cell being moved around as it grows. That's how the mushroom sends food to different parts of itself, is through the hyphae. This is some oyster mycelium, oyster mushroom, growing on straw. It looks like a web. And this is on a stick, what mycelium will look like. It's fun to dig in the leaves a little bit. Well, maybe not for everyone. Fun for me, and to find mycelium. And it's sometimes it's really ropey like this. I noticed the mycelium. I have some logs that I inoculated with shiitake mushroom. I noticed that mycelium is pretty thick and ropey. And then sometimes you can see the changes in the wood because of the mycelium. So this is a mushroom growing through the wood called Chlorosiboria. And I like to find these pieces of wood. They're really pretty. They're used often in woodworking as well. Mycelium can bioluminesce. So on the left, we have a piece of wood under normal photography, and on the right, with a time-elapsed photography with no light. bioluminescing. This is honey mushroom, Armillaria, and it's bioluminescing on this wood. And then sclerotia is one of the off-ramps, I guess, for the life cycle of mushrooms, or one of the detours, not necessarily an off-ramp. Sclerotia are just these masses of cells that have like extra chitin in them to stay stronger, and they are usually underground. Morel mushrooms have a sclerotia part of their life cycle where they can be underground as sclerotia for years and then suddenly decide that it's time to fruit, and all the energy that was in the sclerotia is sent into fruiting and creating mushrooms. It's one of the reasons that morel mushrooms are really common after a fire, is that they decide that it's time to spread their spores instead of growing through the soil. And it's not that the mushrooms are suddenly there growing in the soil. They were always there, just as sclerotia. And then the fruiting body. I thought this picture was interesting. This is another Helvella, probably Helvella crispa. And someone got the spores coming off of the Helvella mushroom in the photograph, and I thought that was a good photograph. So just to recap, we go from spore to hyphae to a network of mycelium, often sclerotia as a detour, and then to the fruiting body that drops spores. So that's the life cycle of a mushroom. But what do mushrooms do? Here they grow through cement, apparently, but they eat wood, they feed plants, they fix carbon in soils, which means that they basically pull carbon or help plants pull carbon out of the air and turn it into soil. Spread the love, which is basically they provide food for everyone, and then they grow all over. Eating the wood means saprobic. Saprobe is something that eats wood. Here on the left is called brown rot, and on the right is called white rot. So wood is made up of a big molecule called lignocellulose, and I believe brown rot eats the lignin and leaves the cellulose, and white rot eats the cellulose and doesn't really eat as much lignin, but I might be wrong on that. So you can see, some mushrooms you can identify by the type of rot that is in the wood that they're growing on. So this is a brown rotting mushroom, and this is a white rotting mushroom. Like chicken of the woods, I believe, is a brown rot mushroom. So that's one way to see it. And then they feed plants. On the left is this, I want to say it's maybe a pine. And there are roots of this plant that go right about to here and right about to here. And then the rest is mycelium. I don't know if you can see my, I think you can see my cursor. But the rest is mycelium. It's they grow throughout the soil, and they're actually able to use acid to dissolve phosphorus that's in the soil and give the phosphorus to the plant. They can either wrap around plant roots or actually grow right into the plant root on a cellular level, and then provide these resources, water, phosphorus, zinc, even nitrogen. But in exchange, the mushroom gets sugar from the plant because the plant makes sugar in its leaves. So mushroom gets sugar, and then the plant gets water and resources. The next step is that mushrooms act with different things in the soil to make something called glomalin, which is a pretty stable protein, basically, that has lots of carbon in it. And this is responsible for a huge amount of carbon sequestration by grasslands in soils. There's basically no soil. It's basically dust without mushrooms, being part of this cycle. So there's a lot of interesting research showing how soils around the world can take carbon out of the air and potentially mitigate climate change. And then one of the coolest things that mushrooms do is provide a network underground for plants, different mushrooms, for the whole forest basically to communicate. They've proven this. They've seen it in action. And they've seen plants mount like a protective response when when someone starts eating another plant nearby, based off of some of the information passed through this network, this fungal network, basically. And then they provide food for all sorts of creatures, including humans. Mushrooms can grow pretty much anywhere. They are almost a requirement before a lot of plants can grow in a spot. I think 70 or 80% of plants are mycorrhizal with mushrooms. They can. A lot of people ask if it's okay to pick mushrooms. Yes, it's like picking an apple. It doesn't hurt the apple tree. So the excuse me, the mushroom is the fruiting body of this huge network underground, and it wants to spread spores. And when you pick it, you're spreading the spores. So you're helping the mushroom, or you're helping the fungi. And they've run a experiment where they picked everything in a section of the forest, and then they picked nothing in another section of the forest, and they do counts every year of how many mushrooms are coming up, and it doesn't change. It doesn't matter really whether everything is picked or everything is left. If you go out in September, there are so many mushrooms that I think it would be difficult to pick more than is eaten by slugs or squirrels or whatever is eating them. I've seen more that just fall over rotten than have been eaten. So there's truly, I think, enough for everyone, and no need to shame people for picking mushrooms. Now we're finally to the meat of it. Just a moment. Excuse me, Michigan mushrooms. This is my son. We like to go hunting for mushrooms as a family. Like to take lots of pictures. So I'm breaking this up into spring Michigan mushrooms, those that occur during the summer, during the fall, during the winter, and then some that you should use caution when you find something that looks like it. In spring, We're going to be looking at Morchella or Morel, Verpa, Gyromitra, Neophavelus alveolaris, and Serioporus squamosus. These are mushrooms that I always find in Michigan in the spring. So here is a Morchella. Everyone knows it, or most people know it. It's really hard for me to see them, honestly. I find a lot more of these other mushrooms during the spring than I do morels. But once you find them, I think you get an eye for them. I mean, I have found them, but still don't have the eye, so maybe that's not true. But some people find buckets of them. They're delicious. This is a Verpa. It looks a lot like a morel, but it has this basically a cap, the stem underneath here, and believe it or not, it tastes similar to a morel. I believe it is also edible. And then Gyromitra. I put this up because it's got 4 different species that we find in Michigan. We just found Gyromitra caroliniana in Michigan. Well, I'm sure it had been found before, but we finally, a friend of mine found it and sequenced it and confirmed that it was in Michigan. It's a very large, beefsteak or false morel, along with Gyromitra corfii is really similar to Gyromitra caroliniana, edible. And then I also see a lot of Gyromitra brunea, which usually has like 2 or 3 folds basically around the cap, 2 or 3 major folds as a way to distinguish it. And then the Jaramich Esculenta group usually has a dark cap, and these are toxic if not boiled a couple times. I believe boil once, change the water, boil again, and then cook them in a pan or whatever. And boiling actually in a lot of mushrooms enhances the flavor because it's breaking down the chitin in the cell walls. So don't be scared about your mushroom's going to taste terrible if you boil it first. And then actually all the Gyromitras and the Verpa and Morel have at least small amounts of the same toxin that's in Jaramich Esculenta. So they all should be cooked, but the amount is very small and is cooked off by ordinary preparation for these 3 Gyromitra species, the Brunea, corfii, and caroliniana. It's just you need the extra step because there's a lot more of it in Esculenta. Neophavelus alveolaris, I find this every spring, growing off of sticks, basically, or small branches. It's really pretty underneath. It has like a hexagon structure, pore structure. Typically, it is even more orange than these photos. These must be a little old. And I like to find them and photograph them. I think they're pretty. I believe they're also edible. And then Dryad saddle, Serioporus squamosus. These are edible. They also have the hexagonal pore structure underneath. These are edible if you can, get your teeth into them or get a knife through them. So when they get really large, they get really hard to cut and chew. So they're best when you find them when, before they're enormous. So get out in the woods. Some summer species that I like to find, I always find chicken of the woods, always find lobster mushroom, always find chanterelle, and sometimes find trumpet, craterellus. But I wanted to provide that because I know a lot of people find them. This is chicken of the woods, Latoporus sulphureus. There are two versions. One is really yellow underneath, and that's sulphureus. And then there's one that's white underneath, but still orange on top, and that is Latoporus. I think it's Cincinnatius, something like that. I don't know why it's escaping me. I find this on really, really rotten stumps. Unfortunately, this beautiful carving is being rotted out by the chicken of the woods mushroom. But usually I find them on stumps that are more well rotted than this. I mean, maybe inside of this carving is really, really rotten. I don't know. But usually really, really pretty rotted wood I find these on. They are pretty tasty. People like to cut them into strips, batter them, and deep fry them. Eat them like chicken. Then this is a Hypomyces lactiflorum. So this is lobster mushroom. I usually find these in clearings and not like in the deep woods. Find them in meadows or at the edge of the woods often. And this is actually one mushroom eating another, but the end result is delicious. Gotta go back. So Hypomyces actually infects and eats either a Lactarious mushroom or a Russula mushroom, and it ends up turning a white or a red capped mushroom with white gills into this orange mushroom. It's pretty unmistakable, pretty hard to misidentify. So it's a good mushroom for beginners, I think. You have to really clean it to get the dirt out of it a lot of times before you cook it. I like it with lots of butter, garlic, and thyme in a pan. I think that's a good preparation for it. These are chanterelles. Actually, I find a lot of different chanterelle species. I usually find them a lot darker than this, or a lot more orange, more like vibrantly orange than this. I find these in late summer, usually. They have veins, technically, instead of gills, which is one of the differentiating characteristics from some toxic mushrooms that I'll talk about in a minute. Veins, like they branch off of each other, and gills are usually just straight and don't branch off of each other. They're usually running down the stipe, or the stalk, whatever you want to call it. So they run down the stipe as veins, and that's how you tell that it's a chanterelle. Trumpet mushroom, pretty hard to misidentify, I think, especially because of the color. There aren't that many black mushrooms. It looks like a trumpet. It's another interesting summer mushroom. And then in the fall, the fall is the best time to find mushrooms. Specifically, I think from the last week of September until late October is the best time. I think this picture was from Midwest Mushroom Camp at Circle Pines Center. It's in Delton, Michigan, south of Grand Rapids. And this was a few years ago, 2019, I think, in the last week of September. And we found just tables and tables and tables filled like this of mushrooms to identify, look at. talk about. So it was a lot of fun. But we'll talk about Russula species or Russula because we always see those. Grifollo Frondosa, which is hen of the woods. Hericium species, also called lion's mane. Lactarious species, and then Lepista nuda, or it used to be called Clutospina nuda, which is something I find every year. So this is a red Russula. They are everywhere in early fall, moving into later fall, and even I find them a lot in summer too, actually. But these are one of the ways to identify mushrooms is actually to taste and spit. Because there's chitin in the cell walls, you don't get a whole lot of toxicity, a whole lot of poison when you taste and spit, even a poisonous mushroom. And so it's a well recognized identification technique. But if you taste and spit a red Russula in Michigan, you're going to either get something that's pretty flavorless or something that tastes like horseradish times 10 that's really spicy. So it's a fun trick fun trick to pull on people if they're annoying you, I guess. This is another Russula. Sometimes they come in purple, sometimes they come in green, just different species of Russula. It's hard to identify them down to species when they're all red. They are either Russula emetica, which is usually spicy, or one of another 10 species or so. I want to say this is Russula parvovirens or something like that, but it could be something else. Hen of the woods, Grifollo frondosa. This is hard to misidentify. I always find it growing around a large living oak, like a very large oak that looks like it's doing really well, but maybe not because of this mushroom. It's also called Maitake. It's purported to have medicinal properties. It is a very common edible. With all edible mushrooms, I recommend taking a small amount first to see if you are allergic, because there are different compounds that mushrooms make that can cause an allergic response. So take a bite, wait, even if you're certain of your identification, and then cook the rest a day later, so that you know that you're not allergic. Some more hen of the woods, Grifollo frondosa. And then moving into late fall, early winter, I find Hericium or lion's mane. It comes in a couple different varieties. I think this one is Hericium coralloides, and this one is Hericium erinaceus. They're both edible. Taste like, I think people make imitation crab cakes out of them because they have like a seafood type taste. This is also medicinal. They've done some research showing that it improves the myelin sheath on nerve cells and help protect the myelin sheath, or at least some components in it too. I'm not sure if they've done, like the gold star double-blind studies to see if it has a therapeutic effect. But components of it protect the myelin sheath. And then Lactarious species. This is Lactarious Indigo. It's not very common, but its cousins are common. All the Lactarious mushrooms are common, or other Lactarious mushrooms are common in Michigan. Usually they start out one color and then end up another color. So Lactarious Indigo starts out blue and ends up a little more green. See where it's bruised here, it turns green, but where it's not bruised, it's all blue. They also bruise different colors, and then they secrete a milky substance from the gills, or if you like cut it open, which is why it's called Lactarious. I find a lot of orange Lactarious that turn green, and those are in the Lactarious deliciosus group. They are not as delicious as the name implies, but they're okay. And then this is Lepista nuda or Clutospina nuda. So I find these a lot in around October in Michigan. They are a very pretty purple mushroom. I believe they have a white spore print. Check me on that before you eat them. The true Clutospina nuda or Lepista nuda is going to smell like frozen orange juice concentrate and is like a European relation. And then there are new species that are very closely related, new meaning North American, basically, that are very closely related that look the same but don't smell like frozen orange juice concentrate. And that's one of the things about mushrooms is that there's always new discoveries. I'll talk about that in a minute. And then some winter mushrooms, some things that you could find right now. We're going to talk about Sarcomixa serratina, oysters, turkey tails, Photopsis Spatulina, which is a birch polypore, and then Flamilano Velotipes. This is Sarcomixa. It's also called like an olive green dapperling, I think. It looks like an oyster mushroom, except it's always got this like olive cap, darker cap, yellow around the young part of the gills or around the gills. I find it early winter. I actually have never eaten this. I don't know if it's edible, but it's something that I'm excited to find when all other mushrooms have shot their shot and are done. I'm excited to find this. And then I do find a lot of oyster mushrooms, which are edible during the winter, during or after a warm up in winter. These will pop out. I was driving down the street in downtown Grand Rapids, maybe not downtown, but one of the neighborhoods, Crestmore, I think. And I just saw these popping out of a tree. They don't mind urban environments. They don't mind the cold weather. So I do find a lot of winter oysters. Turkey tail, it doesn't really fruit during the winter, but they're strong and robust enough that they stay on sticks, and you can just find them out in the woods anytime, pretty much, including the winter. These are being researched for cancer treatment. I wouldn't hang my hat on replacing any cancer drugs with this, but it's promising, I suppose. They are not something that I would eat. I would make a tea out of them if I wanted to extract the medicinal qualities. This is Photopsis Spatulina. I always find these on fallen or almost fallen birch logs. They rot birchwood almost exclusively. This is used as is so hard that it has been used as a strop for sharpening knives. They can be hard to get off of the log, and they are also supposedly medicinal. I'm not sure what the qualities or the results one can expect are. And then Flamilano Velotipes. These have a furry or velvety stipe or stalk, especially down towards the bottom. They look like they're covered in gelatin or water a lot of the time. They have a cap that looks really wet. They are edible, but you have to use caution because there are lookalikes that can fruit around the same time that are toxic. And so we'll look at some of the cautionary mushrooms. On your left is one of the angels, Amanita. Somewhere in section Phalloideae. So I always tell people to use caution for any Amanita, and I'll go over some of the characteristics of Amanita. And then Galarano Marginata, Envelotus Eludins, Chlorophyllum molybdites, and then white Clutosiboid mushrooms. So Amanita, Galarina, and the Clutosiboid are potentially deadly, and then Envelotus and Chlorophyllum are going to make you feel really, really bad. So they're not potentially deadly, but so on my right is actually an edible Amanita grisette, I believe, and on my left or on your left are some Amanita mushrooms in section Amanita, which usually have like Amanita Muscaria, they usually have a couple components in them, ibotenic acid and muscimol, and those can be extracted with multiple boilings, and then you can eat these after a couple boilings, a couple changes of water, and then if you don't extract them, you can have a bad time. Either they have a psychoactive effect, or they can provide a toxin that hurts your brain. It's bad for your brain. But Amanita usually have a skirt. Often they have these warts on top of the cap, and then they usually have either a bulbous base or a cup with a little bit of this veil around them. Here's an Amanita Muscaria. In Michigan, they're all Amanita Muscaria variation Gessoii. And then these two are some of the deadly ones. This is, I believe, Amanita Magnavolare. It has this cup-like base, very pronounced, strong skirt, thick skirt, white cap. And then this is in that section, but not Magnavolare because it doesn't have that strong skirt and that very bulbous base. But it is also one of the deadly Amanitas. Let me go back, actually. The toxin in Amanita that you want to look out for. And actually in the next mushroom, Galarina, will hurt your liver. Or actually there are two different toxins. One, I think, attacks the liver directly, and the other stops your RNA from transcripting. And this is Galarano Marginata. It looks a little bit like Flamilana, which we talked about earlier. It's easy to misidentify as Flamilana. It can grow in the winter, just like Flamilana. So you have to differentiate by a spore print. This will have like I think a rusty brown or orange spore print, and then the Flamilana has a white spore print. Okay, Envelotus Eludins, also called jack-o'-lantern. People mistake these for honey mushrooms a lot. Sometimes they mistake them for chanterelles. These actually have gills running down the stipe instead of the veins of a chanterelle. Sometimes these cause foxfire or bioluminescence, so you can find them at night bioluminescing away in the forest, which is one of the reasons they're called jack-o'-lantern. They're a very pretty mushroom to photograph and to observe, but you don't want to eat it. You'll have a bad time for sure. Just to show the difference, here's the chanterelle. So this is Envelotus with the gills, chanterelle with the veins. And then on the left is a chlorophyllum molybdites, and on the right is an edible mushroom called Leucogerus americanus. They're really similar, as you can see. The way to differentiate between these two is chlorophyllum will start turning green on the gills and will always drop a green spore print. Green means you're going to have a bad time if you eat it. And then these are some white Clutospina mushrooms. I think this is Clutospina rivulosa. I find a lot of big, sort of generalized, nondescript white mushrooms in the woods, and I just stay away from eating them because I'm not good enough at identifying whether they're Clutospina or something edible. These have something in them called muscarine, which is a toxin that affects your heart. All right, wrapping up. Mushrooms. There's a lot of contribution right now from citizen scientists because the field is wide open. So I'm not a mycologist. I have a lot of friends who are not mycologists, but a lot of them have contributed. So go to your local library, learn about mushrooms there. I have to plug the library because I'm presenting on behalf of KDL. And then mushroomexpert.com is good. That's Michael Kuo's website. He's a English professor, not a mycologist either, but he's still a published author. He's discovered a few new species of Morel. Alan Rockefeller has found tons of new mushrooms. And this is Counterculture Labs, a picture from his labs, basically, or where he does a lot of his work. He's not a mycologist either, but he's described tons of new species and has contributed a lot to mycology. MushroomObserver.org is a great place to upload your photos and get an ID. Same with iNaturalist.org. Upload your photos to get an ID. Follow Facebook Mycological Word of the Day if you want to learn more about some of the words that, some of the Latin binomials, why they're named what they're named. And then in an emergency and in an emergency only, post on the Facebook Poisons Help group So this is if you misidentify something, you start feeling funny, post photos there. Or if someone you know eats something that is potentially toxic, post photos there and they can help you identify it. Tips for mushroom identification: post the picture in the habitat, post underneath the mushroom. You need the base, especially for Amanita mushrooms, so you have to dig it out a little bit. And then cross section, cut it in half. There are a lot of boletes in Michigan that if you cut it, it'll turn bright blue really quickly, and that's an identifying feature. Special thanks to Anthony Recca. This is Anthony for all of the photos that he helped provide. Alan Rockefeller agreed to provide a bunch of photos. Anthony Michael Blowers agreed to provide the indigo, Lactarious Indigo photos, and then the Michigan Mycological Society as well. You can find me if you have questions for me at amateskin@gmail.com. Legacy Polyculture on Facebook. Mateskon Law. If you do have a legal question, I'm happy to answer that. And then in the fall, I intend to attend Midwest Mushroom Camp, COVID permitting, and then all of the forests all the time. I'm always out looking. So hope to see you out there. Thank you. Okay. Oh, Sarah, I don't think I can hear you.
You're right. My mic was muted, so I didn't say anything while you were presenting. I saw some fabulous photos, and you actually helped me identify some things I found in my yard while I was raking leaves last fall.
Perfect.
This is great information, and I'm really excited to be able to check out some of those websites you recommended and share my own photos because I found some very interesting things. We appreciate you helping so much with this broadcast and look forward to having you in person. Hopefully spring, summer, fall this year.
Sure.
Thank you so much and have a wonderful day.
Thank you.
Don't forget to check out more streaming opportunities at kdl.org/streaming or follow us at kdl.org/events. Thank you.